So now I welcome you all in the today's webinar on graphene quantum dots, an indispensable future material for biomedical and optoelectronic applications. So today's webinar will be delivered by Dr. Sachin Kadian. Before start of the webinar, I would like to give a brief introduction of Dr. Kadian. So, Kadian, Dr. Kadian, can I start with your introduction?
Sure,
Okay.
please go ahead.
Okay, so Dr. Sachin Kadian is a postdoctoral research assistant at the School of Material Engineering, Purdue University, USA. Prior to this, he was a DST serve overseas visiting doctoral fellow at the Department of Electrical and Computer Engineering, University of Alberta, Edmonton, Canada, supported by Government of India and University of Alberta. He earned his PhD degree in Polymer and Process Engineering from Indian Institute of Technology, Roorkee, India in 2021. He received his MTech in Biomedical Engineering from National Institute of Technology, Kurukshetra in 2016 and BTech in Electronics and Communication Engineering from University Institute of Engineering and Technology, Kurukshetra, India in 2014. He has co-authored over 30 publications in reputed SCI journals, international conferences, as well as book chapters. Based on his PhD research work, he has received 8 awards and recognitions such as Outstanding Article featured in Top 50 Most Popular Article, Best Paper Award, 3 Minute Thesis Award, SERP, OVDF, etc. His co-authored work has also been highlighted in Nature India as glowing biosensor to detect an aggressive lung cancer. His current research interest includes the synthesis and characterization of quantum dots, graphene, sulfur, carbon, perovskites, and zinc oxides, nanomaterials, nanocomposites, and their application in biosensor, energy storage, photovoltaic, chemosensors, bioimaging, and antibacterial agents. So now I would like to invite Dr. Kadian to share his slides and start with the lecture. Thank you. So, Dr. Kadian, the floor is all yours.
Thank you, Sumit, for the introduction. And can you see my screen now?
Yes, it's visible.
Okay, so I think now it's in full screen. Just a minute. Yeah,
Yeah, it's in full.
so okay. So thank you, Sumit, for the introduction. And I would like to thank all the team members of IEEE Nanotechnology Council Student Chapter IIT Indore and the advisory board for inviting me on this platform for the webinar. Today, I'm going to talk about graphene quantum dot and indispensable future material for biomedical and optoelectronic application. So currently, I'm working as a postdoctoral research assistant at Burke Nanotechnology Center, Purdue University, West Lafayette, USA. So before I do so, I would like to uh, acknowledge all the funding agencies, Ministry of Education, Department of Science and Technology, SERB, and my alma mater, IIT Roorkee, and the University of Alberta, NIT Kurukshetra, CSIR NPL, and my current institute, Purdue University, and my PhD supervisor, Professor Gaurav Manik, and UL Alberta supervisor, Professor Karthik Shankar, and seniors, lab mates, and collaborators, friends, and family members who helped me in my research work and making my research journey more happier. So now this is the outline of my talk. And before going to the Graphene quantum dots, I would make all the participants familiar to the quantum dots, and then I will go to graphene quantum dot, properties of graphene quantum dots, and synthesis of GQDs and its application. And before concluding, I will discuss our contribution in this field that we have tried to make some some kind of good GQDs, doped GQDs, and they were also been applied in several applications. So let's start with the quantum dots. So the first question that arises here is, what are the quantum dots? So quantum dots are tiny particles having size in the range of 2 to 10 nanometers. They can absorb light and convert it into the electricity, and they can uh, convert electricity into different colors and light emitting diodes and displays, as we see in every day of our life. Or if we can say, if we have any screen, like if I'm giving presentation, then it is also a part of these things. So next, they can convert the color of radiation into another, and they can also be known as radiation converter. So the interesting thing is that how they do so, like how they can convert this light into different colors or in different like electricity or LED. Or so the main thing is when we excite an electron with external energy, it gets that energy and excited and visit to the high energy level. Once it will stay for very few seconds, and then it will come back to its original state. While it would be coming back, it is releasing some kind of light, or we can say photon with energy, and that energy is deciding factor of the particular light or color or wavelength. So now, why how they are showing the different colors? So behind that, the main role of the gap of these two energy bands. So they this if the gap would be higher, the size would of the quantum dots would be lower, and they would give more like blue shift kind of color. So now the next question that comes in mind, that how quantum dots, why quantum dots only give this property? Why the bulk material is not giving such kind of properties? So in bulk material, we cannot see the quantum confinement effect and quantized electronic state. So as the size decrease, the particles the size of the particle is too small to be the comparable to wavelength of electron. And if you would see that um, the band gap in bulk material is narrower as compared to the quantum dots, and as the size of quantum dot decrease, the band gap increases. So as the size decreases and this gap increases, this signs, or this shows a quantized or discretized electronic state. Because if you would see here, there is all the energy states are in continuous manner, but they all are in discretized manner. So we see all these effects because of their size and number of atoms present in their particular size of or material. So it's all because of these two things, two phenomena that occurs at quantum level, that is quantum confinement effect, and that the width of the bands is confined, and the electronic states are quantized. So I think everyone get some basic idea what is quantum dots and why we are talking about quantum dots. So now I will talk about the emergence of GQDs and their properties and their applications and our contribution. So as we all know, the carbon is the most abundant material on this planet, and it's have been the immense interest for the researchers and scientists since last 100 years. So and the main property of carbon, that it can be bonded in several ways, and because of that, it lots of allotropes came into story of carbon. So as we know, the physiochemical properties and the conducting properties of diamond and graphite, they have been the most used allotrope of carbon. But with the invention of fluorine in 1985, the era of synthetic allotrope of carbon began, and several kind of synthetic uh, allotropes of carbon came into account, and such as graphene, carbon nanotubes, and now GQDs. So since the discovery of graphene, due to their interesting properties, such as it is the thinnest, flexible, and transparent material, and excellent conductor of heat, it has been used in several applications, and but due to some limitations that it has zero band gap, low aqueous solubility, toxic qualities, it has been it has been not used in few of biomedical or some of the optoelectronic applications. But to overcome these limitations, researchers worked on that, and they regulated their structure, and they concluded that if this large graphene sheet is censored into small pieces, this would give some interesting characteristics. And in this direction, Graphene Quantum Dot came into the story of carbon allotrope, and these GQDs exhibit some additional virtues, including size-dependent band gap, excellent aqueous solubility. As we discussed, that as the size decrease, the band gap increase. So if we will make these GQDs of different size, then the band gap would automatically be tunable. So this is an excellent property that they exhibit, and they are good. They are showing good biocompatibility as well, and they are fluorescent. So they are showing different good merits and as compared to graphene. And GQDs are one or a few layer of graphene with size ranging from 2 to 20 nanometers. So these are the properties of GQDs, such as tunable band gap, as we discussed earlier, and they can be synthesized easily, high chemical stability, and they are able to interact with biomolecules due to lots of functional group on the surface. And as the size can be varied, they can show multiple fluorescence, and they are the part of graphene. They are soft graphene sheet. They are a good conductor. So, and they show high photostability against photobleaching. So they are replacing the traditional dyes or semiconducting quantum dots as well. So now the thing is that the ability to tailor the desired physical, optical, and chemical properties also make them more attractive for several applications. But the question here that comes that how we can synthesize these Griffin quantum dots. So we can synthesize them very easily, and there are the two approaches. One is top-down approach, and the other one is bottom-up. So the top-down approach is refers to the breakdown of large graphene sheet or graphite or we would say CNTs or carbon fibers. So if you would see in this Dong et al. Have used carbon black for synthesis of single layer and multi-layer GQDs through chemical oxidation. They have used HNO3, and they have prepared two type of GQDs, multi-layer and single layer. And similarly, Chenatel used electrochemical cutting of graphene sheet, which was prepared using a CVD process, and they have used it for ion sensing. And then Luetel prepared this white fluorescent GQDs using graphite, and they have used sonication in strong acids. So as you know, each coin has two sides, so they have advantage and disadvantage as well. The advantage is that they can be prepared using like graphite or graphene material, which is easily available. But the thing that make them like that limits their use, this process is they are using highly toxic acids, and their removal of these acids is not so easy. So to replace this process, then few bottom approach came into account, and bottom up approach relies on the fusion of small carbon containing compounds such as citric acid and glucose, and then citric acid and urea, mainly those all the carbon containing compound which are having the uh, uh, carbon and we can say aromatic rings. So they would create some graphitic structure. As you can see, that Dong et al. Used the tuning of carbonization degree, that if they stop the carbonization in between, they prepared GQDs, and they completed it, and they prepared the carbonization, sorry, graphene oxide. So if we see that this process is quite easy, and I think it is taking highly 10 to 12 minutes to prepare GQDs. And similarly, Tang et al. Have used microwave hydrothermal process. So in this also they have used glucose as the raw material. And as compared to uh, the top-down approach, the bottom-up approach are quite easier, and they are opening the opportunities for doping of these quantum dots with another heteroatom or some other metal or something, whatever we want to tune our chemical or physical or biological properties. So they have been used much as compared to the top-down approach. But yeah, both methods are interesting. And now because they're easy to synthesize, and they can be functionalized with different functional groups, and they can be doped with different heteroatoms. They have been used in several applications. So now I'm going to discuss applications of GQDs. So first I will talk about biomedical applications. So in this the first one is biosensing. This is our collaborative work, which was performed by Kalkal et al. And this work was also highlighted on the Nature India. So if you would see that they have used amine functionalized nitrogen doped GQDs, and this blue color is showing nitrogen, and this one is oxygen and hydrogen and carbon. So if you would see, the fluorescence of anti-NSE amine and GQD was quite high, and as they come into the contact with gold nanoparticle, as gold nanoparticle are the good acceptor, so the energy transferred by the GQDs has been accepted by the gold nanoparticles, and once they come into contact with the antigen of NSC, the fluorescence uh, started regaining, or we can say we came into the own state with the fluorescence of amine functionalized NGQD. So in this way, we quantized, and we came to know that this NGQD can be used as fluorescent biosensor for the small cell lung cancer biomarker detection, as they are fluorescent material and they are replacing the traditional dyes. So they have also been used as bioimaging agent. And if you would see, Zhang et al. Have used these insulin conjugated GQDs as imaging probe for live tracking of insulin receptor in 3T3L adipocytes. If you can see here, these insulin conjugated GQDs have been attached to the insulin receptor, and they can be tracked throughout the process because they are glowing in green in color. So because of their good photo stability and very interesting fluorescence, they are replacing now the traditional dyes, and they are showing good bioimaging application. So further, due to lots of functional sites on their surface and a good biocompatibility, they have also been used for drug delivery application. And in this, Kodade et al. Used the nitrogen doped graphene quantum dots, and they loaded it with the MTX drug, and then finally they used it for cancer treatment. So they have used the MTX NGQD nanocarriers for drug delivery application. So due to these fascinating properties the GQD possess, they have been used in several applications. And as a part of next application, people have, like as we know that AZ silver nanoparticles and any like graphene or CNT or any other electron of carbon, the composite of these both always showed excellent antibacterial activity. So with this, people have also used GQDs for preparation of a new nanocomposite, and they have shown interesting antibacterial activity. You can see in the same image that have been treated and untreated with P. aeruginosa bacterial cells, and they showed excellent antibacterial activity as well. So further, uh, as these GQDs have several sites or containing oxygen functional groups, so these all this all GQDs can generate singlet oxygen, which can be generated through the light irradiation. And if you would see this, these are the tumor of mouse, and in this they have used GQDs plus light irradiation. In this they have used GQDs only, and in this they have used light irradiation only. So they came to know that GQDs with blue light, they can destroy this tumor because they are generating singlet oxygen, and which is like interesting for killing the tumor or cancer cell. So they have also been used as photodynamic therapy. So beyond that, they have also been used for optoelectronic application because of their excellent optical and electronic properties, their tunable band gap. They can be used as light emitting diodes, solar cells, and photodetectors, supercapacitor, etc. So while we were knowing this all, so since if I would say like 5 years back, I started this journey, and we came to know that GQDs are showing interesting properties. But the one main research gap which we found, and we came to know that the fluorescence quantum yield of GQDs is still poor than the traditional semiconductor quantum dots and the dyes. So we started working on that part, and we thought to work on the doping of sulfur atom into the graphene quantum dots, and we started to see what would be the effect of heteroatom doping on the fluorescence and quantum yield of the graphene quantum dots. So with this, we started our journey, and we formulated 4 different type of sulfur doped graphene quantum dots, because we were starting from the initial part, and we kept the citric acid part constant, and we moved the MPA, that is mercaptoproponic acid, the source of sulfur, that we varied, and we saw that the 300 microliter 10 minutes and 200 degrees Celsius temperature was quite good enough for highly fluorescent sulfur quantum dots. And we the fluorescence in quantum yield also showed excellent results, that the quantum yield of GQDs was reported 11.5%, and we reported our SGQDs with 57.11% as the which is very high as compared to GQD. Then we characterized all these differently prepared SGQDs, and we came to know that it is showing excellent photoluminescence, and which is excitation independent spectra also. So cutting the long story in short, that we characterized all these uh, um, 4 type of HGQDs, and we came to know that they were of 4 nanometer size, and this is the quantum yield that we claimed that our synthesized HGQD showed almost 57% quantum yield, so which was very high as compared to the GQDs. So once we synthesized this, we came to we started using them for different applications, and we first we saw the sensing application. So this is the schematic representation of synthetic procedure of SGQD and sensitive detection of picric acid. In this, you can see the bare SGQDs were very fluorescent in blue color. You can see the curve as well. But as they come into contact with picric acid, they lose their fluorescence property due to the inner filter effect, as they made a ground state complex, and they quenched as we were increasing the concentration of picric acid. So these were the parameter optimization studies, and upon like um, uh, optimal conditions, it was found that the fluorescence intensity of SGQDs was quenching linearly as soon as we were incorporating or providing the picric acid in the solution. And you can also see the SGQD-PA complex in these were the samples which we saw that, yeah, there is some visible color change, as well as there was some fluorescence quenching. and they have shown good sensitivity in the presence of all same type of compounds, which are also nitroaromatic compound. But the thing is that why they were not showing any effect to the other compounds was because they were not able to show any kind of inner filter effect. So the main mechanism was dependent on inner filter effect. Other than this, there are two, three other mechanism, if you would say, like fret or energy transfer, as we discussed in the earlier uh, 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 that biosensor application. So, so in this process or in this part, inner filter effect was very active, and that's why it was able to sense the picric acid. And moving to the next application, we saw the antibacterial application of our prepared SGQD. So to assess that, we prepared AgSGQD nanocomposite, and we saw that our composite was successfully synthesized. As you can see, these small graphene quantum dots have been attached to these AGNPs. These are bare AGNP, and these are bare SGQDs, and this is the composite. So when we performed the antibacterial activities and MIC calculation, we came to know that the our synthesized AgSGQD was showing excellent antibacterial activity as compared to SGQD and AGNP. So we proved it, and we saw that yeah, our SGQD is working as stabilizing agent and the reducing agent for AGNP, because it was only one step method, and we done with the antibacterial application. Next, we as the fluorescence of these SGQDs was quite high as compared to GQDs. We used them for bioimaging application for the detection of FR positive cancer cells. So if you would see, we used folic acid in situ for the synthesis of FASGQD, and this FASGQD attached to the FR receptors, and then it have been taken by the cells through endocytosis process, and they were the hypothesis is showing that they should glow in the FR positive cancer cells. So first we confirmed the synthesis, and it can be seen that the a peak of FA were also present in FASGQD, and these are not limited to these characterization, but we did lots of other characterization as well. But we are just showing that FASGQD was synthesized successfully. And then we came to our main study, that we were doing the bioimaging of FR positive and negative cells. So FR positive are cancer cells, and these were the normal cells. So if you would see, after the incubation of 2 hours, there were some kind of blue fluorescence these cells were showing. But after four, these were quite good in color, and they were showing that yeah, SCQD has been taken by the cells, and we can say that yeah, our synthesized process was working quite well. And if you would see, these are the FR negative cells, as they were not interacting much with the FSGQD. So none of the SGQD was taken up by these cells, and then we can differentiate cancer cells and non-cancerous cells. So this work was selected by the editors of Microchemica Acta as the outstanding article. Those 8 articles were selected in uh, by the Microchemica Acta editors in 2020. And then moving to the next part, we were also curious to see the optoelectronic properties of our prepared SGQDs. Then we incorporated these SGQDs in the perovskite films, and then we saw the electron transfer properties, how it is reacting and how it is reacting on the surface. So we first we characterized the surface morphology, and we came to know that SGQDs are also providing the center of nucleation to the perovskite to make good growth. And then we came to know that it is also affecting the absorption spectra of pristine perovskite. So conclusively, we found that these SGQDs can also be used in optoelectronic applications, because we were in between of these experiments. We didn't go for any like LEDs or solar cell application, but yeah, we proved that it can be used as electron enhancer in these perovskite thin films, and that would be useful for the optoelectronic applications. So in summary, we can say that GQD's research is still at an infant stage and has great potential to become the most attractive material of graphene family for various applications, including biosensor, bioimaging, drug delivery, antimicrobial, photodynamic, photovoltaic, LEDs, photodetector, photocatalysis, supercapacitor, etc. And if more accuracy and preciseness in the synthesis of size and shape control GQDs with high quantum yield and active edge functional group is required to achieve new milestone in the several real-world biomedical and optoelectronic applications. So this is all about GQDs. That still there are some requirements to improve the properties of these GQDs, and they can be truly used in biomedical and optoelectronic application in near future. So these are the contact details, and you can ask any queries. Thank you so much for your attention.
Thank you, Dr. Kadian, for such a wonderful talk. Now, participant floor is open for the questions. You can unmute yourself and ask your question directly to the speaker.
Thank you very much, sir, for a wonderful talk. I have few queries.
Yeah.
Is there any difference between carbon dot and graphene quantum dot?
Yeah, there is some difference between carbon dot and graphene quantum dots. It's mainly that has been reported is about its photoluminescence, and carbon dots majorly shows excitation dependent photoluminescence spectra, and graphene quantum dots are showing some type of independent, as I showed in the talk, that these GQDs are excitation independent, and also they are different in some properties that if we would say carbon dots can be synthesized by only carbon containing compounds. Those are, if we would say, um, like this one, the living organisms and the plants and these. So people have synthesized several type of carbon dots using the leaves and these things. So yeah, there is a difference, but still people are in confusion that how we can say that they are graphene quantum dot or carbon dots. But I would say that the TEM image can differentiate this about their structure or their gap in the fringe widths. So there is difference, I would say.
Yes, sir. Thank you, sir. My another question is that as you have talked about sulfur dope graphene quantum dot.
Yeah,
So why you have focused only about sulfur atom? Why not other
yeah.
atoms just, instead of sulfur?
Yeah, thank you for this question. The thing is that when we started, at that moment, nitrogen and boron were almost explored, but because we were reading lots of paper about the nitrogen doped GQDs, and as I showed you in the presentation also, that NGQDs have already been prepared by people, and I don't found any systematic investigation of sulfur doping, because it's also a kind of heteroatom and it showed good results. So we started that we should explore something new which has not been explored earlier. So that's why we use sulfur as the doping agent.
Yes, sir. My question actually is related with as you have talked about AG functionalization, AG.
Right.
But
Yeah,
you have talked about in when you talked about doping, so you have taken sulfur atom. Can be taken metal atom as a doped atom instead of
yeah,
sulfur
we can.
AgAU?
Yes, few reports are there that they have used metals in the doping as well. So they have also used. And in if I would say, in my study, that was not say silver doping. That was the synthesis of silver nanoparticles using sulfur doped graphene quantum dot as reducing and stabilizing agent. So in this, the if you would see, just a minute, let me go back. And Yes. If you would see, these are the silver nanoparticles, and
Yes, sir.
over there the SGQDs has been decorated like
Yes, sir.
this. So there is no doping. They are just attached through AGS bonding.
Yeah, so that's
So
why I'm saying the functionalization of sulfur graphene quantum dot on AU atom actually. Here, na?
yeah, it is possible. Yeah, it is possible, and I think it's already in the literature.
Okay,
If you
that's
would search AU SGQD,
yes, sir.
then it would be there for some application, I guess.
Okay, thank you very much, sir, for your
Yeah.
response.
Yes.
So any other questions from participants?
Thank you,
Yeah, hello,
Dr.
sir.
Thank you, Dr. Sachin, for such a wonderful talk.
Mhm. I have one
Uh,
question.
Yes, please go ahead.
Yeah, so sir, like as you said that the graphene carbon dots emits various colors. So while
Mhm.
choosing precursor, do you target any particular color? Or it's just the outcome of the chosen precursor.
No, as I said, this color is only dependent on the size. If you would see here, I showed earlier in this slide that yes, here. If you would see here, it's the color of fluorescence is dependent on the size of the quantum dots and the gap between these energy bands. So if you are going to make a red fluorescent SGQDs or sorry GQDs, then the size is the only thing that you can control, and you can get the fluorescence color as you need. So this is the good quality of these GQDs, that these quantum dots, not GQDs, that you can control everything according to your synthesis protocol. So the raw material is, I don't think so raw material is giving you any kind of like help in your color of fluorescence.
Okay, thanks,
Yeah,
sir.
yeah.
Yeah, I have a question. One question I would like to ask with Dr. Sachin Kadian. Can I
Please.
ask him?
Yeah, sure, please.
Yeah. I just wanted to know the graphene quantum dot which you have synthesized. What is the effective BT surface area of this material? What you synthesized over there?
Can you repeat your question, please?
What is the effective surface area? Because whenever you're talking about the from application point of view, let's
Yeah.
take an example of supercapacitor devices. When you're going to fabricate it, its effective surface area should be very large, very higher surface area is required over there, isn't it? So I just wanted to know what is the effective surface area of this graphene quantum dot which you have synthesized over there.
Yes, your question is quite interesting, but I have never worked in the supercapacitor application part. So I
Right.
can answer you through the email, but because I never even read a paper on supercapacitor application of GQD. So
Okay.
this might be an interesting application according to the properties of GQDs. So I have to
Right.
check it before answering this.
Okay. Yeah, I have
Yeah.
another question that
Yes, please.
nowadays people are synthesizing this graphene quantum dot as well as graphene material, nanomaterial using biomass. So
Yes.
do you have any idea about it, that how to synthesize this graphene quantum dot and its physical implications from application point of view? Whether it is giving a at par results as we synthesize using Hermos method, well established Hermos methods, or whatever another methods are existing over there.
No, the only thing is that if you are using the biomass that is having the carbon compound in that, and you are making it either the hydrothermal or any of the method, the only thing is about your properties or characteristic, what kind of characteristic it is showing. Like if you would say few of those materials are not showing uh, good fluorescence and quantum yield. I have also synthesized these graphene quantum dots, or you can say carbon dots, using uh, uh, the leaves of pomegranate, and those were not so exciting, so we didn't report any such kind of results. But yeah, people are preparing these kind of biomass based graphene quantum dots or graphene. So they all dependent on the properties, what are kind of properties they are exhibiting. So I think uh, instead of those biomass, if we are planning to go for real-time application, then there should be some good type of raw material that can result in good we can say photoluminescence and your quantum yields. So that is very important for few of applications. And if they are looking for only GQDs for some like uh, other kind of applications, if we would say, if they are showing electronic properties in good manner and they are not showing any kind of fluorescence, then yeah, they are useful for those applications. So we cannot say that they are not useful. Yeah, they are useful, but they are dependent on the application part.
Yeah. Actually, that is the point of concern that whenever you're going to use biomass, so what about the reproducibility of the material? And from application point of view, how you're going to report it? Because different climatic change is going to give different biomass, and whenever you're going to report it, so that is the point of concern from my point of view.
Yeah.
Okay, thank you.
Thank you.
Yes, sir, I have a question.
Please.
Sir, what is the wetting property of these graphene containers? Means is it durable? Like after some days, is it stable, sir?
Yeah, it is stable. It is highly stable.
Actually, I prepared some carbon containers from biomass.
Okay.
Those are showing the high fluorescence properties as usual, but it is with some time it is wetting, sir. It is sticking to the glass.
Okay.
So what will be the reason there?
They are sticking to the gloves. What was your question?
Glass substrate. Means it is
Okay,
like some becoming watery.
okay, so there must be sticky. The biomass you are using is I think sticky, or it may also be dependent on the pH. You can check the pH of your as prepared carbon quantum dots, and then this kind of problem might be resolved. So it can be due to the pH also. I think the pH of your solution would be in acidic or something. So you should check it once.
7.2, I think, sir.
Yes. So you can check it while you change it, and then you can also check it. And which type of biomass you are using?
Lemon peel.
Okay. So so I think the your raw material is itself very sticky. I think so. So
Okay.
yeah, this is the reason, because you are using a sticky material. So
Okay.
this is
Thank
the
you.
reason. Yeah.
Sir, as you
Thank you,
said, like the size of the carbon dots affect the color. So is it the case with carbon dots also?
It would be with every type of quantum dots, because this I showed for the general quantum dots. This is for all type of quantum dots. Either it's semiconductor quantum dots, or they are metal nanocrystals like AU nanocrystals. They are also fluorescent. So they all dependent on the size, because the size is controlling the band gap, and the band gap is providing the color.
Okay, sir.
Yeah.
Dr. Sachin. May I ask a question?
Yeah, sure. Please go ahead.
Sir, as you have shown the top-down approach, can you go to that slide?
Yeah, please, sure. Yes.
Yeah, so here are several methods, like more of the chemical oxidation. Chemical oxidation or electrochemical oxidations, right? As you have
Yes.
also with sulfur doped your GQDs, right?
Yes.
Sir, I am interested or I am intrigued to know whether you have more of the graphene or graphene sheets are already reported as toxic, right? You might have already
Yes, you are
came
right.
to know. So
Graphene
the
seeds. Graphene seeds are the large graphene seeds are, yes.
yes, sir. So my curiosity is like, I mean, in which antibacterial, like which bacteria you have tested? It is gram positive or gram negative bacteria,
Yeah, both we tested, gram positive and negative. Both we
and it
tested.
was showing the antibacterial properties in both to the same extent or the different kind of
No, it
activity.
was different. If you would see, it was different in both the bacterias. If you would see, yes. See, the one is positive and one is negative. So AGSGQD was showing 35 microgram per ml MIC, and this was 70. So I
So,
think, yeah, it is, yeah.
so sir, that's why, sir, my further curiosity says that if there is a difference in both of the bacteria strains, suppose gram positive and gram negative, there might
Right.
be some interaction or some kind of disruption with quantum dots, graphene quantum dots, as well as silver particles, because silver is also known to be toxic to or having the antimicrobial properties. So
Right, right.
did you follow this to any further extent? Whether what was the mechanism or what was the basis of this kind of interaction? Is there any?
These SEM images are we collected these SEM images, and we came to know that this nanocomposite is disrupting its physical structure or its walls, and no further conclusion was we were not able to make any other conclusion. That's why we went for SEM images, and we saw that the treated ones were showing some damage to their structure. If you would see the zoomed part, and
Yes, sir.
this is the untreated one.
Yes, sir.
So and we also saw the like biocompatibility study. So
Yes.
AGNP were showing, obviously they are toxic, then they were showing higher as compared to this AG-SGQD nanocomposite. And SGQD were also showing very minor, or we can say no like uh, this toxicity. So
Okay.
I think there is something that is happening, but that is still need to elucidate. But AG and its composite with the graphitic structure, graphene, or we can say CNTs or GQDs or SGQDs, they are showing synergistic effect because of some sharp edge of the graphitic material and shape and size of silver and charge of silver nanoparticles.
Might be, sir. Might be, because until we explore, until we get to the, I mean, the whole crux, we will not know what is the
Yes,
process.
yes.
One more thing, sir. When
So
you
yeah, sure.
showed the cancer bioimaging through your graphene quantum nanodots,
Mhm.
could you please go to that slide?
Yes, please.
Yes, sir. So here also, that like citric acid, and you have done with the folic acid, right? And
Yes, yes.
and the cancer cells have very high folic acid receptors,
Receptors, right, right.
right? So, sir, did you try the internalization of these with folic acid attached to the carbon nanodots, or you just synthesize the folic acid nanodots?
See, if you will see this process, so we have used citric acid and mercaptoproponic acid and this one folic acid. Okay,
Yes.
and we used them in situ, and we synthesized because we controlled our reaction, and we stopped the reaction in between, and we came to know that few of the folic acid were were structured in the GQD, and few were outside uh, uh, the GQD structure. So as we performed the experiment, so it was just a hypothesis that these FASGQDs, which we have synthesized, would interact with the FR positive cells, and they interacted, and then only they showed this kind of blue color because they were blue in fluorescence, these SGQDs, and they showed this effect, but there was no FR positive in the FR negative cells. Then these cells didn't show any kind of fluorescence after 4 hours as well. So we prepared this FA-SGQD in situ, not a external attachment of FA on SGQDs.
Okay, thank you very much, sir. Sir,
Yeah.
I just think one small, I mean, kind of suggestion, if you could take it down.
Yeah, sure. Please,
I mean,
please. Yeah.
I will definitely, I mean, I would definitely like to see this work go ahead because, sir, as folate receptors are very common in the animal model, or let's say most of the organisms, living organisms or
Right.
cellular models.
Mhm.
So if you could just devise these further and upgrade them, whether they can distinguish between suppose folate receptors are many times expressed in the cancerous cells, many times than
Yes.
the normal cell. So
Yes.
if we hypothesize, we will find that it may the quantum dots will also, your graphene quantum dots will also get inside the normal cells. But if the
Yes.
intensity will vary to a very much extent, like the folate receptors
Yes.
that are expressed in the
Yes.
cancer cells, they might actually create a very high difference for the bioimaging
Yes.
and real-time bioimaging kind of thing.
Right.
So that
Right.
you might also be working on that, and that you might also go for a further study. But that this is very,
Yeah. Thank.
very interesting results, sir, and I really
Yes.
congratulate on that to you.
Yeah. Thank you. So yeah, I take your suggestion positively, and I understand that all the cells have some FR over there. They are not totally FR free, but yeah, they are deficient in FR like features, and cancer cells are enriched with the FR surface. So that's
Yes,
why they are just showing. And
yes,
yes, your point is right. Then we can use it for real application as well. So
yes,
it
sir.
was just to laboratory purpose only. Yeah. Thank you for your suggestion.
Thank you very much, sir, for your insightful
Yeah.
talk
Thank
and
you.
your knowledge.
Yeah. Thank you.
Any other question from participants? Okay, there are two questions in the chat box.
Someone is speaking. Yeah, please.
So may I ask one question?
Sure, please, please.
So I have one general question, that what does FWHM represent in the fluorescence spectra? Is it related to particle size?
Which one? Can you repeat?
What does FWHM, full width at half maxima, represent in the PL spectra? So it's general question.
Which peer? It's general, not related to the site, like this one slide.
So, no, sir. It's not
Okay,
related to anything.
okay. So in the PL spectra, that is full width. What was your question? Can you repeat it?
So what does this FWHM represent? Is it directly related to the particle size or something else?
Uh, FWHM in PL, we cannot say that it is particle size, related to particle size. But yeah, the emission of PL can relate that what could be the particle size. I don't think so that FWHM is related to the particle size.
Okay, sir. I have synthesized some quantum dots. It shows fluorescence in the blue region. But when I
Uh-huh.
got for zeta sizer experiment, it shows
Okay.
200 nanometer size.
Okay. So
So,
if you are aware about that zeta sizer, or I think we can say DLS. So it is dynamic light scattering. So it is showing hydrodynamic size of the particles, but it's not the accurate size. And you might have a high concentration, maybe in that solution in which you were making your DLS experiment. So I would suggest go for the very light solution, and then you can find your real, maybe. But for true size, if you want to know, then I would suggest go for TM. This DLS can give you the estimated size, but it would not be the true one. I would say that.
okay, sir. Thank you, sir.
Sure.
So one more question. If we have synthesized quantum dots that were in the liquid,
Yes.
but we want it in the powder form, how can we
Yes.
go for?
You can go for lyophilization. It is very easy. Any of the solution which is in water, you can go for lyophilization process. So
Which process?
yeah, lyophilization, freeze drying. If you can search on Google that what is freeze drying or lyophilization. So in this, the water molecule directly evaporated from its frozen state to that one. and the only powder would remain there. So yeah, this is the problem with many of the students, and it was problem with me as well in the earlier phase. But I also came to know that freeze drying process is the only one which can give us the powder of these GQDs.
Okay, thank you, sir. Thank you very much.
Yeah. Yes.
Thank you. Thank you, everyone. There is one more question in chat box, sir.
Please, yeah.
This is from Richa. Difference between graphene QCDs and RGO.
Okay. So RGO is reduced graphene oxide, and graphene quantum dots are like uh, oxygen enriched graphene sheets, if you would say. And the RGO could be of your like larger sheets, but GQDs would be in smaller in size. If you would say that they would be a few hundred or less than 100 nanometers. So this is the difference, and they are also a graphitic structure, but they are, as the name suggests, they are reduced graphene oxides. Okay, so the oxygen moieties over there are very less, and in GQDs they are enriched in the oxygen functional group like OH, COH, etc. So this is the main difference.
So one more question participant is asking.
Yeah, please.
What about Maxine in biomedical application? It is
Uh,
in chat box.
so as I'm not so aware about Maxine, but yeah, Maxine is also going to be used in few of biosensing applications. One of my collaborators is currently working in the composite with Maxine, and it is a kind of I think acceptor. So they are using it, but I think it's little toxic. I cannot comment on this because I'm not working on the Maxine. So it would not be right to comment on this thing. Yeah.
Thank you, sir, for
Yeah.
such a wonderful talk, such a knowledgeable talk.
Yeah.
I think there are no questions further from the participants. So
Please.
I think there are no questions. So let's thank our speaker again. Uh,
Thank you, Vidyasagar. Thank you, Deepak. Thank you so much. Two participants are still commenting in the chat box, so I was just reading.
Thank you, sir, for such a knowledgeable talk.
Thank you, Sumit.
So now, if okay, if there are no questions from participants, thank you. I would like to thank our sponsors for which Hyper Vacuum, Jagatech Corporation, Excel Instrument, Rudraksha Technology, Optimized Solution, Streamline, Mansa Technology for supporting our IEEE NTC student chapter. So I would like to thank you, Dr. Sachin Kadian, for the lecture. Thank you, sir.
Thank you, Sumit. Thank you so much for uh, your patience and listening me. And I'm thankful to all the team members, Pawan, Mayank, and all other persons who are involved in this wonderful work. You are really like providing a good platform to all the researchers for interacting with each other. And then I would also thank the advisory board And yeah, thank you so much
Thank
for
you, sir.
inviting me. Yeah,
Thanks for such
I
a
enjoyed this talk. Okay. Bye, Sumit. Thank you so much.